Hi, my name's Chloe Marino, and my project is on Xenopus as a model organism to study autism spectrum disorder. So, ASD arises from a variety of environmental and genetic abnormalities in development. This graphic here is showing all the different ways that ASD can arise convergently, meaning that the same phenotype is expressed, which is an ASD phenotype, from a variety of different origins. Some of the factors that we know cause ASD are teratogen exposure during development in utero, genetically heritable mutations, and prenatal infection. One common thing that we see across all of these different factors is that they in some way affect the development of the central nervous system because ASD is a neurodevelopmental disorder. So whether that be affecting development in utero before the baby is born or after the baby is born, early in development, in some way these factors that cause ASD are actually affecting the development of the brain and how certain connections are made in the brain. Some of the traits used to diagnose ASD, they're pretty various, and there's lots of debate going on about how ASD is diagnosed right now, but I chose some of the most basic ones, which are social communication abnormalities, issues with motor skills, sensitivity to stimulation, and repetitive behaviors. Some comorbidities with ASD that are common include hydrocephaly, as well as epilepsy, and in severe cases these can be really debilitating to the individual or fatal, which is why it's really important to research ASD. And also it's just important because it's a really interesting neurodevelopmental disorder, and we don't really know too much about it. So why Xenopus? Well, they are an ideal vertebrate model for early development and morphogenesis, and that's because they're an amphibian model. Amphibians have been used in developmental biology research for a very long time. They're pretty robust as compared to mammalian models because they can basically just withstand a lot more crazy experiments and manipulations that you can do to them, which is really helpful for studying developmental biology. They're also if you think about it, frogs, they develop in water. So Xenopus, they develop just openly in water. They have clear membranes surrounding them, so you can see their development really easily. It's pretty visible, whereas with mammals, they have to be in utero, so it's kind of hard to see what's going on during development. And with birds, they're covered by a shell, so again, it's really hard for that visibility. So Xenopus is a really good mechanism to study like actual cell movement and behavior and morphogenesis in the early embryo. So some of the ways we use Xenopus are to study cell biology, biochemistry, genetics, biophysics, and here are two examples of kinds of experiments that you can do with Xenopus. So microinjection is a big one, microinjecting RNA and then seeing what phenotype that leads to, as well as chemical treatments where you treat the water that the tadpole is growing in with a certain kind of chemical and see how that affects growth and development. So the new research that I'm going to be talking about in the following slides basically uses a lot of different kinds of methods, the ones that I included up above actually, to holistically examine ASD from like a broader perspective so that we can see how it affects vertebrate development from a broader perspective, which might be more beneficial. So the current amphibian models use in vivo expression of ASD risk genes from humans to examine their effects on morphogenesis. So a recent genetic screening analysis was done that identified 71 different genes in humans that are associated with ASD when mutated. So it was found that a lot of individuals with ASD had mutations in these specific 71 genes, and many of these genes are homologous with Xenopus. So my focal paper, which was written by Wilsey, was super interesting because it was the first time that they actually did a parallel in vivo analysis of ASD genes in an amphibian model. So what they did was they expressed 10 of the most associated ASD risk mutations in humans in Xenopus tropicalis, and they just observed the phenotype to kind of see what happened because this had never really been done before. It was a really helpful way of convergently inducing ASD to make an ASD model. So convergently, as I mentioned before, they were using different kinds of mutations on different kinds of genes, but they led they wanted to see what kinds of phenotypes they did. Did they lead to the same phenotype? Because these are all associated with ASD, but in the tadpole, do they actually lead to the exact same thing? So they used a CRISPR-Cas9 complex in order to do this, which worked by they injected half of the embryo with the CRISPR-Cas9 complex that was associated with their specific gene that they wanted to mutate, and they only injected half of the embryo so that when the embryo developed into a tadpole, only half of that whole side of the embryo divided by the midline or the spinal cord was expressing the mutation, so had a loss of function mutation in the specific ASD risk gene, while the other half of the organism was just the control, so that they could compare both sides of the tadpole at the same time, which is a very reliable model rather than having different kinds of organisms which could have, subtle phenotypic differences that would be hard to differentiate. So once they grew these tadpoles to a later stage, they assessed them for their phenotypic differences. So what they found was that the telencephalon size, which is a part of the brain in the tadpole, is varied in the ASD risk mutant tadpoles. So here's an image of all of the some of the different mutant telencephalons, and you can see that all of the ASD risk gene mutants are different than the controls significantly. And here's a better depiction of that. You can see that 4 of those ASD risk genes actually increased the brain size, while 6 of them decreased the brain size. And in order to prove that this CRISPR method of creating an ASD phenotype worked, what they did was they injected a functioning human version of the gene into the CRISPR mutant, and the CRISPR mutant has a loss of function mutation in that gene. So when they injected the functioning human gene, that actually rescued the phenotype, as you can see from the difference between F and G, which is pretty interesting. So here's a more quantitative visual of that happening. So once injected with the functioning human gene, the CRISPR loss of function mutant was able to rescue its phenotype. So they also conducted another study that actually found that estrogen reduced the ASD-like phenotype in the ASD mutant tadpoles. So they conducted a drug screening that found estrogen was a suppressor of the ASD phenotype, and then they went ahead and tested to see if that would actually phenotypically restore the control phenotype in an ASD mutant. And they found that ASD risk mutant tadpoles were actually able to recover their phenotype when they were treated with estrogen. Another study, which was done by McCluskey et al., it's a new publication, it's actually about to come out, and she's actually working with a similar kind of model using CRISPR-Cas9, but she's looking at the enteric nervous system development. So the enteric nervous system develops via early neural cells. It happens super early in development. And what these neural cells do is they're specifically neural crest cells, and they migrate from the top of the embryo down into the gut to establish and form the gut in the early embryo. And what they found was that the CRISPR-Cas9 mutants that were ASD mutants, like I had mentioned before, done in the same way as before, when they actually tracked their neural crest cell migration to form the enteric nervous system, their migration was greatly inhibited, and they actually weren't able to properly develop, or they developed abnormally, those neural crest cells, and where they ended up developed very abnormally. So that's pretty interesting, because there's been a link shown, correlation shown between gut dysmotility and ASD in humans, but there's not really any work that's been done on the genetics behind why, and this could be why. So they conducted a real gut motility assay with these tadpoles, to actually confirm that that lack of proper neural crest cell migration actually showed that there was gut dysmotility. And they found that there was. This actually was true, and they did that by feeding these mutated tadpoles fluorescent beads, and then they let the tadpoles release their stool, and then they caught their stool and they sampled it, and they found and imaged and counted the number of fluorescent beads that the tadpoles had processed through their digestive system. And they found that the ASD mutant tadpoles actually had really, really poor digestion. They couldn't really let any of the fluorescent beads through, whereas controls did pretty normally. So, to summarize, an ASD phenotype in a model organism can be generated in numerous different ways, as it also can be generated in numerous different ways in humans. So research can really benefit from using a convergent approach, which means studying the higher level phenotype of ASD, which might not be rescued by direct molecular interception, but by modulation of neurogenesis, so from a higher level. With something that happens convergently, it might be more effective to study things from, the neurogenesis perspective and the broader perspective, rather than looking at all of the individual mechanisms that 71 different genes might, produce. So future studies could continue to utilize this genetic expression to induce an ASD kind of model. It's pretty reliable because we can genetically confirm that this is relevant and actually a pretty good model for ASD, and it also can help us find more factors that might affect its development and how it can manifest. Thank you. Here's my works cited also.